Hi everyone, my name is Mark Schembri. I'm from the University of Queensland in Brisbane, Australia. And today I'm going to tell you about some of our research involving the characterization of a globally disseminated multi-drug resistant uropathogenic E. coli clone. I'd just like to start by thanking Uta Robling for the opportunity to give this presentation. So this is a fascinating story about the emergence of this UPEC or uropathogenic E. coli clone. The clone itself is referred to as sequence type 131 or ST131, and it's undoubtedly the most successful E. coli clone we've ever seen. So just an overview of the presentation that I'm going to give today. I'm going to give you a brief introduction on uropathogenic E. coli or UPEC and introduce you to ST131 and tell you a short snippet of the fascinating story that describes its antibiotic resistance. And then I'm going to tell you a story about that was published recently that describes the tracking of a recurrent UTI caused by ST131 in a single patient. And I'm going to describe to you the genomic epidemiology involved in the history of infection in this patient. So a little bit of background about urinary tract infection or UTI. This is an extremely common infection, especially among women and the elderly, and it's estimated that more than 50% of women will experience a UTI in their lifetime. The dollars associated with treating UTIs are enormous. UTIs account for around 10 million doctor visits every year in the US and around $3.5 billion annually in treatment costs. And therefore, they're a significant burden to the healthcare system of all countries. UTIs are frequently recurrent, and this leads to highly complicated infections. UTIs are essentially an ascending infection where the bacteria will originate from the fecal flora, will colonize the urethra, and then colonize the bladder and the kidneys. UTI can also lead to life-threatening sepsis, and this has a high rate of mortality and morbidity. Uropathogenic E. coli or UPEC are the major cause of UTI, and it's estimated they cause around 75% of all community-associated UTIs. So UPEC have a range of different virulence factors that contribute to their ability to colonize the urinary tract and cause disease. And the most important of these virulence factors are Schembri. And there are two major types of Schembri associated with the ability of UPEC to cause disease in the urinary tract, and they are type 1 Schembri and P. Schembri. I've illustrated type 1 Schembri here. You can see that there are these long hair-like structures that stick out from the surface of the cell. And importantly for this talk, I just need you to remember that these Schembri contain an adhesin at the tip. That adhesin is called FimH, and it's FimH that mediates binding to demenosylated receptors on the surface of epithelial cells. UPEC also produce polysaccharides on the surface, in particular O antigen and capsule, that lead to protection from host immune factors. Many strains produce toxins. They also produce ion acquisition systems that's important for their survival in the urinary tract. They produce flagella that mediate motility. And we are now seeing many UPEC strains containing large multi-drug resistance plasmids. These plasmids can carry anywhere between 10 and 15 antibiotic resistance determinants. And when they move, they can move en masse. And that means that drug resistance can be shared between different strains very easily. So with regards to antibiotic resistance, the largest story surrounding UPEC involves sequence type 131 or ST131. This is a picture describing invasive E. coli resistance to third generation cephalosporins and fluoroquinolones in Europe. So here we have third generation fluoroquinolone resistance, and here we have fluoroquinolone resistance. What we're looking at here over time is a change in color. So the darker the color, or the higher the level of resistance, and you can see that there's very high levels of resistance around 2017 across many parts of Europe. And these regions here in red represent countries whereby where 25 to 50% of all of the E. coli's associated with invasive infection are resistant to the antibiotics that are classes that are indicated. And fascinatingly, much of this story can actually be told by understanding the prevalence and the emergence of E. coli ST131, because E. coli ST131 is the major contributor to this resistance pattern. So what is ST131? It's clearly and without doubt the most successful E. coli clone that we have ever seen. Since 2008, we've seen the simultaneous spread and high prevalence of this clone across the globe. It was first identified through its association with the CTXM15 extended spectrum beta-lactamase, which mediates resistance to oxaminocephalosporins and monobactams. The lineage also has co-resistance to fluoroquinolones, as well as aminoglycosides. And it's a community-associated pathogen, and that means that it causes infections in the community, as well as in hospital and healthcare settings. It's a major cause of UTI and neurosepsis, and it contains a high prevalence of virulence factors. So a few years ago now, my group teamed up with Scott Beatson's team, also at UQ, and we set about to understand the genomic phylogeny of ST131. And we did this by collecting just under 100 isolates from 6 different regions around the globe. And we sequenced these using Illumina technology. And when we examined the sequence of these, the first thing that we noted was that all of the ST131 strains claded obviously within the phylogroup B2 for E. coli, but separately to other UPEC strains that have been characterized in the literature and may be familiar to many of you just by the strain names. So the structure that we observed was or comprised 3 different clades of ST131, as you can see here, that we refer to as clades A, B, and C. And so if I expand this here and we have a look at the distribution of strains, you can see that we have the following pattern, and this pattern that we described back in 2004, or at least this clade structure, still holds true now, despite many, many more hundreds of ST131 having been sequenced. And if we follow this, then we can see that the 3 major clades are described as such, being A, B, and C. Clade A represents fluoroquinolone sensitive ST131. In addition to clade B, these strains are also fluoroquinolone sensitive. And then here we have this dominant lineage represented by clade C that we've divided into 2 subclades, C1 and C2. And these are resistant to fluoroquinolones. And then between these, we have an intermediate group. In addition to this, some of you may be aware in the literature that FIMH typing can be performed. And if you remember, I talked about FIMH and the role of FIMH as a tip adhesion of type 1 Schembri. Well, if we look at the FIMH alleles that are present within each of these clades, we can see that FIMH 41 is associated with clade A strains, FIMH 22 with clade B strains, and FIMH 30 with these clade C strains. And as I said to you, there are 2 subclades here, clade C1 and clade C2, and it's clade C2 that contains the most antibiotic resistant strains within the clade C lineage. So what we did in addition to this, or at least subsequent to this first analysis that we performed, was to perform a beast analysis, which essentially enables us to place a molecular clock alongside each of the strains that we've sequenced. And that enables us to map the emergence of these different lineages. So what you're looking at on this slide here is just the clade B and C lineages. And I just want you to focus really on the clade C lineage. I told you that this lineage here comprises strains that are all resistant to fluoroquinolones. And if we have a look at this, we can see that this lineage emerged from clade B via this intermediate group of strains. But strikingly, we can see here that the development of resistance to fluoroquinolones in this clade C group occurred around 1987. And that was associated with mutations in GRA and PAR-C that have been well described with respect to resistance to fluoroquinolone antibiotics. This work was published a while ago, and in this analysis we also showed that the most likely region of the world that these strains originated from was somewhere in North America. And fascinatingly, if we actually have a look at the use of fluoroquinolones in North America, we can actually see that fluoroquinolones were first introduced around 1986. So the use of fluoroquinolones coincides almost precisely with the emergence of ST131 strains that are resistant to these antibiotics. Fascinatingly also, what we found was that if we looked back within this lineage, we can see that there are various events that describe the acquisition of genomic islands containing different sets of virulence factors, and they predate the emergence of resistance against fluoroquinolones. So what we really think is that ST131 has emerged by stealth. It's gone in the background unnoticed and acquired sets of genes that enhance its fitness and ability to cause disease. And then in around 1987, when this sequence type picked up resistance against fluoroquinolone antibiotics, that emerged its that enabled its massive population expansion, and that's what we're now seeing. So there have been many studies to try and understand why ST131 has been so successful, and we've been involved in many of those ourselves, and we've looked at antibiotic resistance, survival in the intestine, virulence genes, survival in human serum, survival in macrophages. We've looked at different plasmids that ST131 contain, and we've looked at toxin production by way of hemolysin analysis. And all of these studies have shed light onto why ST131 has been successful, but they haven't been able to really answer the fundamental question, and that is, why has this clone of E. coli become so dominant? Why has it been so successful? And how has it managed to spread across the globe so rapidly and become the most common E. coli that causes UTIs and bloodstream infections all around the world? So as part of that, I just want to then tell you this story that we published recently that involves tracking recurrent UTI caused by ST131 in a single patient. And I think it's shed some really important information with regards to the fitness of ST131 for colonization of the gut and the potential likely reservoir of the gut to seed extraintestinal infections. So the story that I'm going to tell you about here is a fantastic collaboration that involved Matt Mulvey at the University of Utah and Scott Beats and Brian Ford at UQ. And the story involved the investigation of a single patient, a Caucasian female. She had a vaginal hysterectomy in 1971, and since that time she suffered frequent UTIs. In 2011, she presented to the University of Utah Hospital at 74 years of age. In 2012, she picked up an ST131 infection. And because of fears of some complications that may originate in this patient, she received a 21-day course of the carbapenem antibiotic ertapenem. She actually remained UTI-free for about 9 months. And remarkably, this is the longest period of time since the hysterectomy that this patient can remember being UTI-free. So that's over a period of around 45 years. And so anybody who makes comment about this disease and its severity really needs to understand how debilitating UTI can be for some patients. In 2013 onwards, the patient suffered frequent recurrent UTIs. She was given multiple different antibiotics, and generally speaking, her UTIs resolved with antibiotic treatment. So we were fortunate enough to be able to obtain isolates from this patient during UTIs over the periods as indicated. So we isolated an E. coli strain in 2012, 13 and 14, and then 2 in 2015. We sequenced these, and I can tell you that they were all ST131 isolates. And in addition to that, we also were able to obtain 25 ST131 isolates from the Utah community that were really important in enabling us to put the distribution of ST131 variants into perspective in this region. I also want to just emphasize again the fact that between 2012 and 2013, this patient underwent a 3-week course of ertapenem, and I'm going to come back to that a little bit later. Okay, so as I mentioned, we sequenced all of these strains, and then because we had such a well-characterized genomic phylogeny of ST131, we were able to integrate these strains into our phylogenetic tree for ST131. And you can see here that in red, these are the patient isolates, and in purple scattered throughout this tree are the isolates that were recovered from the Utah community. And so what this tells us is that there was variation within the Utah community that mapped and fit or at least represented the global diversity of ST131 that we had already identified. So if we expand on this group of ST131 strains, then we can see that the 5 ST131 strains that we identified and which are named here using the year of isolation as a code, you can see that they're extremely closely related. And in fact, there are only 26 core SNPs that separate the strains from isolated in 2013 and 14, which and those from 12 and 15, and each of those within themselves demonstrate some subclustering. If we have a look at the entire genomes, and we were able to sequence the entire genomes by combining Illumina sequencing with PacBio sequencing, then you can see that the genomes are almost identical. There's very little variation. There's an example of a deletion occurring in the 12 and 15 isolates, as is indicated here, and this corresponds to the LUX genomic island. If we have a look at that deletion, then there are some genes involved in ion transport and an antigen 43 autotransporter that were lost. But apart from this, there's no very clear difference in loss or gain of key virulence factors that is linked to the persistence of this strain over the time period that we examined. We were also, because we had complete genomes, able to investigate the plasmids. And what we did find is that the greatest differences between the strains were associated with plasmid diversity. So this is the 2012 isolate. You can see from here that it contains 8 plasmids. And this INC-F plasmid here contains a multi-drug resistance island, is signified by green. You can see that within each of the strains, there is diversity of the plasmids. And you can see that that involves loss of plasmids at different stages. And remarkably also, we can see evidence of movement of the resistance island from one plasmid to another. This is the resistance island here, as I've mentioned, and we were able to show that that exact resistance island had moved onto the INC-I plasmid in this U15A strain. We can see that in a little bit more detail here. The movement of this resistance island is associated with recombination mediated by insertion sequences. I'm not going to go into the detail of this, but we had the exact movement of the island. And you can actually view that directly if we look at the flanking regions of the island, and we can see that the island has moved en masse from one plasmid to another. So at this stage, we were a little bit puzzled as to what was going on with this patient. There's no apparent community reservoir that we're able to identify. We investigated the spouse of the patient, and the spouse's patient tested negative for ST131. So the question of, what was causing these repeat infections of this patient remained. And we started thinking about whether or not the gut could be a reservoir of recurrent UTI in this patient. And one of the things that really made us think about this was the 3-week course of ertapenem that the patient received. We all know that ertapenem is a highly effective antibiotic at treating UTI. It achieves very high concentrations in the urine. However, one of the features of ertapenem is that it's poorly absorbed in the gut. And so we thought, well, maybe this explains why a gut reservoir wasn't disrupted and eliminated by that course of ertapenem antibiotic. So what we did is we collected 2 fecal isolates from the patient that we identified as being ST131, and we sequenced those patients, those isolates. The 2 isolates we chose were selected because they were both resistant to ciprofloxacin, but one was also resistant to trimethoprim, while the other one was sensitive to trimethoprim. So when we sequenced those isolates, we found that they were indeed exactly the same strain as the strains that had been causing recurrent UTI in this patient. There were only 4 core SNPs that separated the 2 fecal strains indicated here and here. And in fact, there's only 1 core SNP that separates the fecal strain from one of the urine isolates cultured in 2015, and only 3 core SNPs that separate the other fecal strain from this urine isolate. And if we move along this lineage, we can see that these fecal strains clustered most closely together with the ST131 strains isolated in 2012 and 2015. So here's a pattern of the plasmid distribution again, just to highlight what has been happening with the diversity of plasmid dynamics, but also then show what's happening with the plasmids in these fecal isolates. So here's our 2012 isolate. You can see it contains this INC-F plasmid with an, sorry about that. We can see that it contains this INC-F plasmid with the antibiotic resistance island, and that island has moved onto this INC-I plasmid in these strains here. These are the 2 fecal strains, 15RA and 15SA, indicating the resistant and sensitive strain with respect to trimethoprim. And you can see that they have different combinations of plasmids as well. And the resistance to trimethoprim is associated with the presence of this antibiotic resistance island. Okay, so we didn't want to stop there. So what we did is, in addition to all of these isolates that we had collected and sequenced to date that I've told you about, we also collected an additional 60 fecal isolates. We collected 30 isolates in 2015 and 30 isolates in 2016, 15 trimethoprim resistant and 15 trimethoprim sensitive from each of these collection time points. And we sequenced all of these and placed them in our genomic phylogeny with respect to ST131 isolates that have been identified from this patient. When we did that, we found that the fecal isolates that were sensitive to trimethoprim all clustered very nice or very closely together. You can see that they are basically identical. There's a couple of outliers here, but we're only talking about 1 or 2 core SNPs that are different between these isolates. In comparison, those isolates that contain that resistance island in the ink-I plasmid show, I guess you could describe as a greater degree of evolution with respect to, or microevolution with respect to changes in their genome. If we have a look at the plasmid distribution between these strains, then we can see this is the ink-F plasmid, this is the ink-I plasmid. You can see that the ink-I plasmid containing that resistance locus is absent from all of these trimethoprim sensitive strains that have been cultured from the fecal reservoir or fecal sample from the patient. And if we look at the distribution of antibiotic resistance genes, where the presence of the genes is indicated in yellow and the absence of the genes is indicated in blue, we can see that the presence and absence of these resistance genes maps exactly together with the presence and absence of the antibiotic resistance island on the respective plasmid. So just as a summary to describe or to give an overview of what I've told you about today, we were able to use whole genome sequencing to characterize ST131 strains causing recurrent UTIs in a single individual over a 5-year period. This patient was persistently colonized by a clonal ST131 lineage that could be identified in the fecal flora, and the ST131 lineage that colonized the gut of this patient is responsible for causing recurrent UTI in the patient. And this reservoir of ST131 that exists in the gut of this patient is able to persist despite repeated antibiotic treatment that the patient underwent over a number of years. And that includes the treatment with carbapenems, and I think I've summarized why we believe that it was likely that the strain was able to survive treatment with ertapenem in the first place, linked to its poor absorption in the gut. I've been able to show you that there's significant plasmid diversity that occurs within the strains that we isolated. There's the coexistence of antibiotic resistance and antibiotic sensitive intestinal isolates, and that occurs despite frequent treatment with antibiotics. I think that this raises the question associated with whether or not we are observing population-wide resistance. And what I mean by that is whether or not the resistant strains can actually create a microenvironment to allow the survival of sensitive strains. There is no doubt that the intestine is the most likely reservoir for ST131 mediated recurrent UTI in this patient. And I think that this certainly does raise the possibility that the intestine is a likely reservoir in many individuals for ST131 mediated disease. I think our study also raises some important questions about sequencing. If we're thinking about sequencing and tracking strains in infected individuals, then it really shows that we probably have to sequence multiple colonies if we want to get a snapshot of the entire diversity, including the plasmid diversity. It also means that there is a reliance on some technologies that enable longer reads from the sequencing in order to be able to track these plasmids. But then also, I think it raises questions about the role of metagenomics as we move forward. So with that, I'd just like to finish up and just thank the people that were involved in the work. In addition to my own group, I really want to thank Matt Mulvey at the University of Utah and his team of researchers that really enabled this study to take place at all. And also to Scott Beasley and Brian Ford for their fantastic contribution towards getting the sequence analysis done. So with that, thank you very much.